All right, let's dive right into this explainer. Today, we're taking a microscopic factory tour to see exactly how life perfectly replicates its most important instructions. Welcome to the Blueprint Factory. Imagine having to type out billions of letters by hand. No backspace, not a single typo allowed, and oh yeah, you've got to finish the entire thing in a matter of hours. That is literally the monumental task a cell faces right before division. How on earth does a single cell perfectly copy its master blueprint before dividing, making absolutely sure the next generation has everything it needs to survive? I mean, it's an absolute marvel of biological engineering. The central dogma of modern biology brilliantly captures the big picture here. DNA is the ultimate blueprint, right? It stores all that vital genetic information, and it absolutely must be reliably passed down to the next generation through what we call vertical inheritance. Usually, information flows from DNA into RNA, which then builds the proteins. But for this explainer, we are hyper-focused on that very top loop, DNA perfectly replicating itself. So, here's our roadmap for the factory tour. We're going to cover 1. The blueprint of life. 2. Setting the stage. 3. Leading and lagging strands. 4. Finishing the job. 5. The rolling circle alternative. And finally, 6. Reading the blueprint. Section 1. The blueprint of life. Understanding the assembly line. Now, to build anything, you need raw materials and a solid construction crew. And our microscopic site requires quite the toolkit. First up, you obviously need the original template DNA. Then, you need the actual building bricks, these specialized nucleotide molecules we call dNTPs. Throw in an RNA primer to act as a starting block, a massive amount of energy, and an elite crew of enzymes, Helicase, Primase, Polymerase, and Ligase. Every single one of these guys has a highly specific, crucial job on the assembly line. Section 2. Setting the stage. The construction phases. What's really fascinating here is that the entire replication process doesn't just happen in some chaotic scramble. It's highly coordinated into 3 distinct sequential phases. First is initiation, where that tight double helix is basically unzipped and opened up. Second is elongation, which is the rapid, high-speed construction of the new DNA strands. And finally, termination, where everything wraps up and the cell properly closes up shop. It's incredible how this plays out differently depending on the organism. Notice how circular prokaryotic chromosomes just have a single origin of replication to kick things off? The replication forks simply move outward in a circle until they meet up on the other side. But eukaryotic chromosomes, they are linear and absolutely massive. So to get the job done efficiently without taking forever, they require multiple origins of replication operating all at once, creating several replication bubbles that expand and eventually just merge together. Section 3. Leading and lagging strands. The directional complication. Okay, so here is where we run into a major mechanical issue right out of the gate. Our main builder, the enzyme DNA polymerase, is strictly a one-way street. It can only build from the 5' end to the 3' end. And because DNA is anti-parallel, this forces the crew to use 2 totally different construction methods simultaneously. The leading strand has it easy. It's synthesized continuously, moving smoothly in the exact same direction as the replication fork. But the lagging strand, it's synthesized discontinuously because it's literally forced to build in the opposite direction of the fork's opening. When you map out the flow of the newly synthesized strands, it's wild. While the leading strand just smoothly follows behind that bulldozer-like helicase unwinding the DNA, the lagging strand is forced to build backward in chunks. As the fork opens up further, new template DNA gets exposed, meaning the lagging strand machinery has to constantly stop, jump back toward the fork, and start building all over again. This step-by-step workaround for the lagging strand is a brilliant molecular dance. First, an enzyme called Primase drops in to create a temporary RNA primer scaffold. Next, DNA polymerase 3 jumps on and builds a short DNA segment, creating these disconnected sections we call Okazaki fragments. Then DNA polymerase I swoops in to swap out that temporary RNA primer for actual DNA. It's an incredibly dynamic, non-stop cycle of start and stop synthesis. And finally, our site welder DNA ligase steps in. Ligase creates those essential phosphodiester bonds, seamlessly stitching those disconnected Okazaki fragments together into one solid, continuous chain. Honestly, without this crucial final step, the lagging strand would just remain this vulnerable series of broken DNA pieces. Section 4. Finishing the job, resolving the ends.
Once the main elongation phase wraps up, the construction crew has to shut down the site correctly. But in eukaryotic cells with linear DNA, there's a unique architectural flaw known as the end replication problem. Basically, the very tips of the chromosomes can't be completely copied by the standard machinery. Enter the enzyme telomerase. Telomerase acts as a highly specialized finishing crew, ensuring the very tips of the chromosomes aren't lost during each round of division. It protects our vital genetic blueprints from slowly degrading over time. And this protection is huge. In fact, telomerase activity is intimately linked to how our own bodies age and how we protect against cancer.
Section 5. The rolling circle alternative, a different strategy. Let's step back for a second and look at an entirely different architectural strategy used by certain microbes. Some plasmids and bacteriophages use what's called rolling circle replication. Instead of opening a big replication bubble, they just nick one strand of the circular DNA. As DNA polymerase adds new nucleotides, it continuously displaces that old nicked strand, just peeling it away to quickly spool off a single new copy of circular DNA. It is a wildly efficient, continuous loop of replication.
Section 6. Reading the blueprint. Entering RNA transcription.
Okay, so now that our factory has perfectly copied the master manual, the cell actually needs to open the book and read the instructions. To read a gene, the transcription enzyme RNA polymerase must first recognize the promoter. Think of the promoter as this giant start here sign located just upstream of a gene. It features highly conserved sequences, specifically the -10 and -35 boxes, which direct the machinery exactly where to dock and begin transcribing a piece of DNA into a working RNA copy. And this leaves us with the ultimate cliffhanger for this explainer. Once we have this temporary mRNA copy, how do the cell's microscopic factories actually decode it to build the incredible complex proteins that keep us alive? Going from a simple string of genetic code to a physical, working protein structure is a whole other miraculous biological factory process. It's an amazing puzzle to ponder as we wrap up our tour of the DNA assembly line.